Hi everyone, my name is Don Shahira Binti Ismail, a final year student of Food Science and Technology from UIT and Kuala Lumpur, would like to present my final year project, a review on antioxidant properties of Stingless bee honey in controlling microbial activity for therapeutic treatments. Let's move on. Introduction. Stingless bee honey consists of antioxidant, antimicrobial, anti-inflammatory, and moisturizing effects which can help to speed up the recovery process in treating burns and wounds, as Stingless bee honey contains powerful phytochemicals, for example, phenolic content, flavonoid content, antibacterial peptides, and biological compounds. On wound and burn areas, Stingless bee honey properties can slow down the oxidative stress, reducing and breaking the free radical chain, as free radicals can lead to antagonistic alteration in cell component and lead to chain reactions, subsequently destroying the cell. Phytochemical compounds in Stingless bee honey also control the microbial activity of Staphylococcus aureus, Staphylococcus xylosis, E. coli, and Pseudomonas aeruginosa, as most of these bacteria are mostly found on skin with detrimental effect such as infections to the wounds and burns, sepsis, and attacking the central nervous system. Problem statement. Stingless bee honey is said to have high wound therapeutic properties, which is 3 times higher compared to honeybee honey due to its natural product with medicinal properties. Total phenolic content, total flavonoid content, humidity, temperature, and sources of plants influence the antioxidant properties and its activities. There are still fewer shreds of evidence regarding the deep relationship between phytochemical compounds and the ability of Stingless bee honey to inhibit lipid peroxidation, also known as oxidative stress, and anti-radical activity. The use of honey-coated treatment can help in reducing dependency on medicine, thus an alternative ways to prevent antibiotic-resistant bacteria. Significance of study. The literature review compiles current studies of antioxidant in Stingless bee honey in controlling microbial activity under therapeutic treatment, which mainly focusing on burns and wounds. Stingless bee honey is scientifically proven to have the potential to lower the microbial activity of gram-positive and gram-negative bacteria. The objective of this review to discuss antioxidant properties in Stingless bee honey in terms of phenolic compounds, flavonoid contents with additional substances through total phenolic compound (TPC), total flavonoid content (TFC), and anti-radical activity assay through FRAP and DPPH assays. Second, to compare the microbial inhibition zone for different kinds of Stingless bee honey species to prevent the progression of diseases in therapeutic treatments, focusing on burn and wounds. Third, to review the effect of Stingless bee honey in wound healing. As for the research questions, first, what are the properties of antioxidant in Stingless bee honey? How the antioxidant properties in Stingless bee honey help to control microbes during wounds and burns healing process? For the second research question, and for the last one, what is the impact of Stingless bee honey in wound healing treatment? Moving on to literature review part. The first one, antioxidant properties of Stingless bee honey. Antioxidant can be divided into two. First, enzymatic antioxidant convert free radicals into more stable and less harmful molecules. Second, non-enzymatic antioxidant will inhibit the formation of free radicals or prevent the damaging chain reaction. Mainly hydroxyl group is capable to donate electron to free radical unpaired valence electron. Reactive oxygen species ROS lead to DNA and collagen damage due to reactive molecule initiating the membranes, lipids, amino acids, and DNA to collapse. For total phenolic content, higher polyphenols compared to South America honeybee honey. It shows that protocatechoic acid PCA and 4-hydroxyphenyl acetate promote cell proliferation and have the ability to scrunch around reactive oxygen and nitrogen. For anti-radical activity, phenolic substances act as antioxidant agents, have redox properties, provide free radical scavenging ability due to the presence of hydroxyl group. The analysis or the assay can be conducted through FYP and DPPH, where highest scavenging activity is for colluted honey at 40 mg per milliliter recorded. As for the total phenolic acid content such as PCA, p-coumaric acid, and gallic acid, based on the table, it shows that the range of TPC is in between 235.28 to 1166.77, while total flavonoid content is in between 101.5 to 7,902 coming from quercetin, carbapenem, and chrysin. Moving on to another subtopic, antimicrobial properties in stingless bee honey, where phytochemical compounds act as antimicrobial too. Flavonoid, phenolic acid, and ester in propolis has high-spectrum antimicrobial effect. Next, the retardation of microbial growth through microbial inhibition concentration (MIC) and microbial bactericidal concentration (MBC). Based on the result, it shows that Geniotrigonal thoracica suppress the growth of Staphylococcus aureus and Pseudomonas aeruginosa at MIC 5% weight over weight and 10% weight over weight. As for Heterotrigonal itama, it suppressed the same pathogenic bacteria at MIC 10% and MBC 20%. When we are comparing with Taiwan honey from sting bee honey from Apis mellifera, sting bee honey exhibit highest MIC and MBC indicate that it is not a good source to inhibit the pathogenic bacteria due to high concentration of honey required compared to stingless bee honey. Next, the morphological alteration of bacterial cells following stingless bee honey treatment. The result can be analyzed through SEM micrograph. As for the gram-negative bacteria such as E. coli and Pseudomonas aeruginosa, resulting cell shrinkage, cell membrane become coarser, detachment of cell envelope due to releasing of endotoxin or beta-lactamase. As for gram-positive bacteria such as Staphylococcus aureus, clumping septated coccoid cell larger due to hydrolytic activity under stingless bee honey treatment. From the table, antioxidant capacity that indicate how much antioxidant activities between colluted honey and tualang honey shows that total antioxidant capacity was the highest for colluted honey at 208.64 and 201.80 micromole Ta per g at 50 micromoles of honey concentration compared to tualang honey. As for the result in table number 4, the inhibition zone of gram-positive and gram-negative bacteria under stingless bee honey treatment. The result shows that Geniotrigonal thoracica suppressed the growth of Staphylococcus aureus and Staphylococcus saulosus at 2.20 cm and 2.10 cm respectively. As for the Meliponine seminigra, it suppressed the growth of Staphylococcus with the inhibition zone of 2.06 cm. Not only that, The inhibition zone of E. coli and Pseudomonas aeruginosa also shows positive result at 1.60 and 1.00 respectively due to stainless bee honeydew honey treatment and also Meliponidae favolineata honey treatment. Another subtopic discussed in this literature review: overview mechanism of antioxidant and antibacterial activity of stingless bee honey. Factors involving antibacterial activity can be seen through number 1, non-parasite component with the presence of flavonoids and phenolic compound as it prevents superparasite free radical from damaging the tissue. Second, high osmolarity of stingless bee honey provide insufficient water for pathogenic microbes to survive. Number 3, low pH value and high free acidity provide unfavorable condition for the pathogenic microbes to survive. Next, mechanism of wound healing with stingless bee honey as it fastens epithelialization, increased angiogenesis, and collagen synthesis. As for the in vitro study of immunomodulatory and antioxidative effect, it shows that stingless bee honey inhibit nuclear translocation and activation of activated beta cell and FP, decreasing in inflammatory mediators such as cyclooxygenase 2 (COX2) and TNF-alpha. Stingless bee honey can neutralize oxidative stress by increasing TPX, SOD, and nitric oxide level. Last but not least, another subtopic that will be discussed is the application of honey-based coating for wound dressing. Honey-based coating function as a preservative and provide high humidity at the sites of the wound. Characteristics such as non-toxic, non-allergenic, smooth application allowing the exchange of water vapor and also oxygen. Additional of nanoparticles such as gold nanoparticles exhibit antibacterial effect by two modes of mechanism. The first one inhibit ATP enzyme synthase, and second use reactive oxygen species through independent mechanism. In conclusion, antioxidant properties of stingless bee honey depend on the availability of phytochemical compounds and biological activity. Phenolic and flavonoid compounds influenced by the types of flowers and the origin of the stingless bee honey, as well as humidity and temperature. The instability of free radical formation and antioxidant level can be stabilized with the hydroxy group as it has redox properties due to high mobility of hydrogen. There are many phenolic compounds present in stingless bee honey. However, PCA or protocateate acid and 4-hydroxyphenylacetic acid are the strongest to scavenge the reactive oxygen species ROS and nitrogen species. Phenolic and flavonoid compounds also contribute towards the antibacterial effect through non-peroxide activity, inhibiting cell phagocytosis of pathogenic bacteria. The retardation of pathogenic bacteria with the greatest inhibition zone of morphology changes due to stingless bee honey treatment. Honey-based coating with additional antibiotic, for example, ampicillin, and the use of nanoparticle from gold and silver effectively treat wounds and burn as it preserve and provide high humidity to the wounded area. As for the recommendation, first, provide stingless bees with multifloral plants for better honey production and to enhance the quality and quantity of phytochemical compounds. It can be through Antigonolaptopus, Orthosippus staminus, Bidon pilosa, and Caudatus sulphurus. Second, limiting the storage period of Stingless bee honey for better effectiveness. Last but not least, only select the same botanical floral species in all regions to narrow the research gap. That's all for me. Thank you.